So, very good afternoon, my dear students. This is Manikandan, Professor and Head Department of Pharmaceutics, Resenmo College of Pharmacy. So, today we are going to discuss about the vaccine drug delivery systems. The topic which was discussed earlier, definitions, advantages, disadvantages, and all. Then see one by one what are the advantages, what are the mechanism of actions, then what are the various route to deliver the vaccines product. These are the just highlight of this topic. Introduction, types of vaccines, delivery of system for vaccines, uptake of antigens, single shot vaccines, mucosal vaccines, drug delivery systems, then transdermal vaccine drug delivery systems, and another quality control test for the vaccine drug delivery systems. Let's see what do you mean by DNA vaccines. DNA vaccines enna? So the basics of DNA vaccines is first reported by the Ulmer et al. In the year of 1993. Then the injection of the DNA into animal does not generate an immune response, but the DNA is expressed to the yield the protein that can stimulate an immune response. Okay, there are composed of bacterial plasmid. The DNA is introduced into the tissue cells that are host to respond to the antigenic materials introduced to the DNA is the lost from the recipient cells and the antigen release. So for example of DNA vaccines, influenza, herpes vaccines, the measles, HIV, Ebola, these are the DNA vaccines. Then recombinant technology DNA vaccines. The recombinant vaccines is developed through the DNA technology. These vaccines are produced by the insertion of genetic material which encoding the antigen that stimulate the immune response. The plasmid DNA is used as a vaccines which is propagated in the bacteria like E. coli. They get isolated and purified into the vaccines. This method is used to prepare a highly pure component of vaccines. Example, the hepatitis B vaccines, the surface antigen from the hepatitis B, the virus is isolated, sequenced, and that is done by the yeast cells. The first recombinant vaccines which was developed by the India by Santa Biotechnology, Hyderabad. Okay, these recombinant vaccine genetic manipulation of the antigen itself it is possible in the hepatitis B. Then there are some miscellaneous vaccines like veterinary vaccines. These are living or dead preparation of microorganisms used to stimulate the active immunity in the animals. Okay, so the vaccines are recommended on the basis of the kind of exposure, the disease, stress, species, and strains of the animals. There are classified killed live subunit vaccines, example anthrax vaccine and foot and mouth disease. Then another one vaccines, there is a peptide vaccines. The first identification of the peptide had a vaccine's potential was demonstrated in the year of 1963 with the plant virus, the tobacco mosaic virus. In this study, chemical isolated hexapeptide fragment from the virus code proteins, okay, which was coupled to bovine serum albumin and used to elect the rabid antibodies which neutralizes the infection. So these peptide vaccine is now widely against the rabies virus, polio virus, and measles virus and influenza virus. The peptides are the poor immunogens and this require the carrier coupling to enhance the immunity. The first, the free peptide are also effective when they are delivered in a small unilamellar. The peptide vaccine are based on the in vitro synthesized peptides of 20 to 30 amino acids. Then the viral vaccines. These are the vaccines, the suspension of viruses or the preparation obtained from the tissues or blood of animals or from the culture of the fetal eggs cells tissue culture. These may be live, inactivated or killed or may be the freeze dried conditions. Okay, polymyelitis vaccines B and rabies vaccines and smallpox vaccines. So what are the mechanisms involved in the vaccine drug delivery systems? To initiate the adaptive immune response, a number of signals are required by the T cells. The signal one is the vaccine derived, the peptide antigen bound to the major histocompatibility class 2, and class L is displayed on the surface of antigen present in the cells. Signal 2 is also known as a co-stimulation, and together with the single one induce the immune response. There are the two different mechanisms. This signal is recorded by the T cells. Then the signal 2 involves the cross-linking of CD28 and other receptors on the T cells by co-stimulatory molecules such as B71 and B72 and other ligands that are expressed by the active pharmaceutical. The signal 3 is provided by the cytokinase, and it is delivered from the ABC to T cells, which is determines the differentiation into effector cell. The both signal 2 and 3 are provided by to the T cells by activated cells DCS. The mature DCS are able to induce T cells the clonal expansion and prime immune response. Then what are the uptake of antigens? The antigens generated by the exogenous and endogenous antigens processing activate the different effector functions. There are the different stages of exogenous antigen uptake. The access of native pathogen to intracellular pathway of degradation. The degradation is limited proteolysis of antigen to peptides. The antigen MHC complex formation, the loading of peptides on the MHC molecules, the antigen presentation, the transport and the expression of the peptide MHC complex on the surface of cells for the recognition of the T cells. So this is the uptake of antigens. There are the two antigens are generated. One is endogenous and another one is exogenous. Then the innovative in the intranasal vaccine drug delivery systems, the dry powder intranasal vaccine delivery, the gel vac technology developed by the Delsite biotechnology, which consists of the dry powder formulations of vaccines with a natural plant, which is the natural plants that derived the acidic polysaccharides materials which is administered into the nasal cavity. So we will deliver the drug in the form of the powder, dry powder form into the intranasal cavity. Okay, the Becton Dickinson dry powder inhaler. This is technology air from the syringe, the barrel rupture, the membrane of the capsules containing the vaccine, which can be propelled into the nasal passage. Then there are some nano carriers for systematic and mucosal vaccine delivery. The primary reason for using the mucosal route of vaccination is the most infectious affect or start from the mucosal surface only. Okay, from the mucosal cavity only. The mucosal vaccines have currently begun investigated using a broad spectrum of nano carrier. The systems such as a multiple emulsion, liposomes, polymeric nanoparticles, dendrimers, and etc. The latest advancement in the vaccine drug delivery, the cancer vaccine. The cancer vaccines are medicines that belongs to the class of substance known as a biological response modifiers. There are the two broad types of cancer vaccines. The preventive vaccines, another one is a treatmentive vaccines. Let's see, the preventive vaccines are intended to prevent the cancer from the developing in healthy people. The FDA approved preventive cancer vaccines in United States, the Gardasil and Cervarix. There are the two approved vaccines by the FDA that protect against the infections by the two types of HPV, type 16 and 18. The treatment vaccines are intended to treat the existing cancer by strengthening the body natural defense against the cancer in the year of 2010, the FDA approves the first cancer treatment vaccines. This is a choice Suplecel T. It was approved by the first cancer vaccines by the metastatic prostatic cancer. These are the latest advancement in the vaccine drug delivery systems. Let's see what are the various evaluation tests for vaccines. There are the usual tests, that is a sterility test. Incubate the media for not less than the 14 days at 30 to 37 in the test for detecting the bacteria, and 20 to 25 degree the test for detecting the fungi. However, the live bacterial vaccines growth of microorganisms from which the vaccines was prepared is permitted. So sterility test is one of the important tests for the vaccine drug delivery system. Safety test. So inject at least 2 healthy susceptible animals. The quantity to be injected in each animal is the twice the appropriate vaccinating dose. Observe the animals for not less than the 7 days. No animals exhibit the abnormal reactions. Okay. So by the safety test we can identify it. Then abnormal toxicity test. Inject 0.5 ml subcutaneously into each of 5 mice and 2 ml intraperitoneally into each of the 2 quina bigs. If the vaccine being examined containing that adjuvant, inject 2 ml of vaccine subcutaneously into the each quina big. Observe all the animals for 7 days. So none of the animals shows the significant local or systemic reaction. If one animal dies or shows the sign of ill health during the observation period, repeat the test. Okay, so none of the animals of the second group dies or shows the sign of ill health, the test may be omitted if the safety test is carried out on the animals of the species of the vaccine is intended. So identification test. Identify the bacterial vaccines can be checked by the precipitation and agglutination reactions. The inactivated viral vaccines are tested by the observation of the specific antibody responses in vaccinated animals, and the live viral vaccines by the neutralization of their cytopathic effect by the specific antisera. Okay, identification test, whether the bacterial are present or not by the way of precipitations or agglutination reactions. Okay, the phenol concentration. The phenol is used as a preservatives in the different types of vaccines. It is the concentration must not more than 0.5% weight by volume. So these are the tests are more important for the verify the evaluation of vaccines. Let's all still the few more topics are there in the vaccine delivery. Let's see in the next class. Thank you.